Okay, everybody, welcome back. So, I'd like to start with just a little bit of a status of where we are in the course of my lecture. So, um, I think it might be obvious already, but I think it's good to point out sort of this explicitly, which I know I haven't done yet, sort of the spirit in which I've organized these lectures. And I think what's really important in my lectures is I'm not trying to lay out any cutting edge theoretical ideas or any fancy mathematical tricks. I think what I'm really trying to show you is how an experimentalist uses some simple theoretical calculations to design experiments. And I told you in my first lecture, that the real one of the objectives for the field and also for my laboratory specifically right now is to really develop synthetic active materials. So we want to make materials where we can control their properties, let's say just mechanical properties to make it simple, by modulating, let's say, the level of activity in the material. And as I've said several times, in order for something to be a material instead of a system, right, you need to have some idea of homogeneity in the system over some wide range of length scales. And you also need to be able to achieve a meaningful steady state in order to do experiments in the lab to characterize them and study them. And so, I've done a lot of work with living systems, as I mentioned earlier, and it's really spectacular what living systems can do with active materials. And I gave you some examples in my first lecture. And on Friday, what we did is we did a survey across a wide range of organisms and quantified the overall level of activity of the organisms. We looked at some famous old data of how the total metabolism, or the total rate at which the organism rejects heat to its environment, scales with the size of the system. But then I said, well, if we're really interested in active materials, let's look at what the metabolic density is. Okay, so last time we talked a lot about what I was calling Q dot. It was the metabolic density. And this is basically the heat generated per unit volume inside of an active system. And that when we looked across biology, we found that Q dot was, in the smallest case, was about a kilowatt per cubic meter. And this is sort of corresponds to large mammals like us. We produce about a kilowatt per cubic meter of heat. Down to bacteria, which produce about a megawatt per cubic meter. Okay, so at some level, like when you looked at this huge log plot I drew, where there was 25 decades of mass, this small range of 3 decades of metabolic density doesn't really seem like that much, and that's true. But when we think about the thing that really got me thinking about these questions was, we're used to look in cell biology and cell mechanics and the processes like cell biophysicists like to look at. We usually look at eukaryotic cells, like the ones I was telling you about, like the ones that Dave Waits' lab was studying, the things that we're made out of. But bacterial cells have metabolic densities that are 3 orders of magnitude higher, right? And so that really suggests to me that there could be some really interesting and different things that occur at such high metabolic densities. And yeah, so I want to build these synthetic systems in the laboratory. And as I wrapped up last time, the question I wanted to ask, if I want to maintain such a huge metabolic rate, I have to keep supplying fuel to keep up with that. I have to reject heat. I have to reject waste. And so the basic question as an experimentalist is, how small do I have to make my system in order for it to have a homogeneous composition and temperature? Okay? And so how small can it be? That's the basic question. How small can the system be? Now, everything would come to a screeching halt if I found that in order for it to have a homogeneous composition in the presence of fluxes, that length scale would be smaller than the structural scale of the material, or comparable to the structural scale of the material, right? If it's a useful active material, this sort of transport limited size has got to be much bigger than the structural scale. And in order to investigate that, I proposed a sort of a very simple, minimal active system. The idea is that we have a spherical domain which partitions a catalyst, okay? So it has some concentration of catalyst that's greater than 0. Outside of that domain, there's no catalyst. And then far away from that reactive domain, we have some reservoir of fuel. So there's some concentration of fuel at infinity. There's some concentration of waste at infinity. And there's some well-defined temperature at infinity. And so what happens here is that inside of this droplet, we imagine that the fuel gets processed by a catalyst to produce waste, okay? Yeah, and so the basic question that we want to ask, we just want to calculate what's the temperature profile in this situation, and what are the concentration profiles of the fuel and the waste, okay? So some very simple questions. And last time I introduced Michaelis-Menten kinetics, just as Ramin reminded you this morning, so I won't go through it again. But the key thing is, and the first thing to point out, well, actually, I won't go through the whole thing again. I will just say, going forward in our calculations, we're going to operate in the limit where the fuel is relatively scarce. It's below the equilibrium constant for the binding of the fuel and the enzyme, or the fuel and the catalyst, rather, to use my terminology, so that the reaction rate, so S is the reaction rate, I'll write down what that is again in a second more precisely, is proportional to the same K-cat that Golostanian just told us about. Km is just the same as Golostanian's K, the concentration of the catalyst times the concentration of the fuel. Okay, so this reaction rate S is the rate at which you're producing waste, and the opposite of the rate at which you are changing or producing fuel, I guess. Yeah, so that's the basic setup that I went over on Friday evening. What I'll do today is we're going to just work through the calculations and figure out how big or how small things need to be. Any questions before I forge ahead? Nope. Okay. So this is a reaction diffusion problem, okay? And it's a relatively simple reaction diffusion problem because it's linear. And you've already seen some reaction diffusion in Professor Golostanian's lectures. And I think, you know, the geometry is new, but I'll still I'll go through the basic physics and outline the derivation and talk about the physical interpretation of the results. So the first part, of course, is the diffusion bit, okay? So we're going to keep track of the diffusion of all 3 species, potentially. And so we have 3 different species, right? We have the fuel, we have the waste, and we have the catalyst. And in general, which is not what we're going to worry about, but it's important to say the general case because I think it could really, the next iteration might require us to look at this, is if you want to look at the mass flux of any individual species, it will actually depend on the gradients, not just of itself, but on the gradients of all the other species. So in more generally, I want to say that the flux of species I depends on the gradient of the concentration of species J, and there's some mobility tensor or diffusion tensor that connects the two. And in particular, this really ties right into what Glycolysis was telling us at the end. You can imagine he was mentioning, well, do enzymes, which is the equivalent to my catalyst, move in gradients of the substrate, which is the fuel, the equivalent of my fuel concentration, right? That was a question that was posed, and in order to capture that in this picture, you would need some sort of cross-diffusion term, okay? But just to keep things simple for now, as an experimentalist, I always like to use, my general philosophy is you always use the simplest theory you can get away with for your data. And right now I have no data, so I just make it as simple as humanly possible, and I just ignore the cross-diffusion, okay? So if you ignore the cross-diffusion, of course, then we just have good old Fick's law for a single component. Okay, great. Right, so that's the driving term here is gradients and the concentration. All right. All set so far? Okay, good. All right, and then we do the next thing. I bet you can all predict the next thing I'm going to do. What are we going to do next? What do you always do after you write down the Fick's law? What? Yeah, you conserve mass, exactly, right? So we conserve the mass, and I think you all know how to do this. But the thing you have to be a little bit more careful with, because we have a reaction now, you can't just take the div and be over with it, right? So what I mean by that is that when we take the time dependence of the concentration of the fuel is not just equal to the divergence of the fuel flux, right? But there's also a sink term associated with the reaction, right? So that's what makes it, epsilon more than good old Fickian diffusion. And then, of course, for the waste, it's the same thing, except you change the sign in front of the source term, because the reaction, of course, is a sink for a fuel and the source for the waste. Okay. Now, one simplifying thing. Since we ignored cross-diffusion, and if we imagine that we start off with the catalyst homogeneously distributed inside of the droplet, there's absolutely no reason why there will be any flux of catalyst or the catalyst will change concentrations. Okay, we don't. The catalyst is perfectly partitioned into this domain. It can't diffuse away, and it doesn't couple to the other fluxes. Okay. Great. So, as I mentioned before, I'm interested in materials, right? So if I care about materials in steady state, I just want to I want to have a uniform concentration. I want to set the time derivatives equal to 0. And then you take this result, you combine it with that result, just like you would normally do in a diffusion equation, except now you have a source or a sink in your diffusion equation, so that the overall reaction rate for the fuel is equal to the Laplacian of the fuel concentration times the diffusion coefficient, and it's minus that for the waste. Okay. So those are the simple governing relationships. Exactly the same like you would normally do, except now you have a source term. Yeah. Yeah, so I mean we do in that it's sort of hidden. It's hidden in S, right? And so we'll put it back in explicitly later. But for now, the catalyst concentration is absolutely in here, right? And all the sort of the molecular details about how the catalyst works are all lumped into S. And one interesting thing to point out is that's actually, it's good, it's a good way, your question help me lay out the structure of the calculation I'm going to do. For, if I'm looking at the steady state of the waste, you can see that the reaction rate is independent of the waste concentration, right? And also the reaction rates, yeah, but it explicitly involves the fuel concentration, and so that really changes the way that the solution, you'll see the steady state solutions change, are qualitatively quite different for the fuel and for the waste. Yeah. Why the catalyst doesn't change? Because I'm forcing it to not change. So if I start off with a why would the catalyst change? First of all, if it could diffuse out of this domain. I'm saying it's not allowed to diffuse out of that domain. If I start off with a homogeneous, and let's just imagine we start off with some homogeneous distribution, there's no reason why it would ever change. And if I start off with something that's not homogeneous, eventually when I go to steady state, it'll be uniform, right? So, I mean, I could actually, it's probably not even, I probably shouldn't have even said that because that was confusing, right? Perhaps because once I impose steady state, I don't need to say that, and it makes sense that there's no driving force to keep it to be anything other than uniform. Any other questions? Okay. All right, so yeah, so now what we're going to do is it's going to turn out that the fuel, when you go through the calculations, is the thing that's going to be the problem. So we're going to look at the fuel first, and so and we're going to be focusing on this case where so let's focus on the fuel. Focus on the fuel concentration profile in. I'll call this the fuel limited reaction rate, right, as I wrote down there, but where phi f is less than km. Okay. Great, so when you do that, the equations that I wrote on the right become as follows, that the Laplacian of the fuel is equal to some stuff in brackets, which is mostly about the catalyst, kcat times the concentration of the catalyst divided by the binding constant of the catalyst of the fuel, but there's one factor here, which is the diffusion coefficient of the fuel times phi f. Okay, so we have this form, functional form, describing how the fuel concentrations change in space. This is a constant. This is a very familiar form. Professor Golostanian almost talked about, he talked about the nonlinear version of this today. Where have we seen this equation before? In electrostatics, right? So this is the Debye-Huckel equation for screened electrostatics. And in the context of electrostatics, we call this constant in front kappa squared, right? And in the electrostatic analogy, right, for those of you that know the electrostatic analogy, this is helpful. If you don't know the electrostatic analogy, sleep for a second, right? Kappa is, kappa we would call the electrostatic screening length, or the Debye length, right? And phi, here our phi f, would just be the electrostatic potential. Yeah, and so the key point to summarize, the solutions to the Debye-Huckel equation is that in contrast to the Laplace equation where potentials fall off like 1 over r from monopolar charge, when you have screened electrostatics, there's an exponential decay of the potential on top of the 1 over r decay that has a characteristic length scale of kappa to the minus 1. Okay. Here, for reasons that will become obvious eventually, that this kappa to the minus 1, it basically does the same thing, but you'll see I like to call it the penetration depth. Penetration, or length, let's call it, instead of depth. And you'll see why. But it turns out the basic idea, right, is there's some far field concentration and of the fuel, and it starts getting consumed as it goes into the sphere. And we'll see that this kappa to inverse is basically how far can the fuel get before it reacts with the catalyst. Okay. Questions, comments about the setup so far? I'm not going to solve it. I'm just going to write down the answer, because you can find the solution in all sorts of places. All right. So here's the solution. There's 2 domains because the concentration of the catalyst is different on the inside and the outside. So inside of the reactive domain, it has the following form. There's one term here. The first term is just the prefactor, which depends on the concentration of the fuel infinitely far away, and a hyperbolic function of the size of the object. But then all the real spatial dependence is here. And as you often see in the electrostatic problems, you have a form of something exponential with the kappa inside over something that looks like 1 over r. Okay, so inside we expect something exponential as well as some slow 1 over r behavior. Okay, outside. So for radii larger than the size of the reactive domain, it looks quite a bit different. Again, it's all proportional to the total fuel, the concentration far away. And then it has this funny little bit. So 1 plus tanh of kappa big R minus kappa big R times 1 over kappa little r. Okay? So for some fixed value of the size of the sphere and the screening, right, there's something that falls off like 1 over r. Okay? So I'll sketch what this solution looks like before I sketch it. Yeah? Oh, that should be, oh, that's supposed to be phi f, sorry. I'm not talking about phi w at all until later on. Yeah, this should all be phi f. Thank you. Now I realize I should have flipped the classroom and asked you what you thought the profiles would look like before I calculated them and drew them, but try to, yeah, anyway. Regret. Okay. So this is what the profiles look like. So I'm going to plot things up as versus r divided the distance from the center divided by the radius of the reactive domain, and then on the vertical axis I have the concentration of the fuel divided by the concentration of the fuel at infinity. Okay. Now and I'm going to put a dashed line here where the distance from the center is equal to the radius, or in other words, the edge of the reactive domain. And now one really important thing in the structure of the solution is that there are really 2 free parameters in the system. There's little r, right? So there's the distance from the center. And the other important, there's a dimensionless group, which is kappa times big R, right? And that and so the profile only depends once you normalize it by the step in the far field, it only depends on this value kappa times big R. And when kappa times big R, when kappa times big R is less than or approximately 0.1, what you find, if this is 1 here, that basically the concentration of fuel is unmodified by the presence of the catalyst, okay? And so the physical interpretation here is that the rate of diffusion for the fuel across this relatively small domain is much higher than the rate of the conversion of fuel into waste, so there's hardly any change to the fuel concentration at all, okay? Where if you go sort of in the opposite limit, let's say if we go to kappa R, I'll make it a different color. Kappa R, no, that's not a different color, that's the same color. Yeah, there we go. Kappa R, let's say greater than or approximately equal to like 50, okay? So that means that we have a really big reactive domain. So you have to get, it turns out that basically the concentration of the fuel is 0, basically right when you get to the boundary, and basically increases like 1 over R. So it increases very slowly from there, okay? So there's sort of two extremes. One extreme where the fuel can go everywhere. The other extreme is where the fuel doesn't make it inside of the droplet at all. And of course, there's stuff in between. And so I'll draw kappa R equal to 10. In between here, kappa R equal to 10. Yeah, so kappa R equals 10. There is still a strong suppression, but slowly varying outside. And then once you get inside, there's a little bit of penetration into the core, towards the core of the reactive domain. Okay, so and most of the all the fuel gets eaten up over some distance, kappa to the -1, because it's the only length scale in the problem. Okay. Another fun way to think about this is to ask what are the reaction profiles? Where is the reaction happening? Okay, so if we plot up the reaction profile, all right, we can write. Remember that the reaction is maybe I erased it. It's proportional to phi c, and it's proportional to phi f. Okay. Outside of here, phi c is equal to 0, so there's no reactions on the outside. And then basically the reaction rate will be just proportional to phi f on the inside. And it's almost obvious, but I think it's worth plotting. Okay, here here's the edge of the droplet. Here is the reaction rate at the edge of the droplet. I mean at the center. Here's the reaction rate everywhere normalized by the reaction rate at the edge. And again, if we try to use the same color, if kappa R is less than or approximately 0.1, you have a perfectly uniform reaction rate inside, which is what we want if we want to make a material. And then let's say we have kappa R equal to 10, then everything is localized over a very narrow band of kappa to the -1. Okay, so really all the reactions look like they're happening at the surface. Okay, super. Okay, so that stuff, I think that's pretty straightforward in lines with things you've already seen in the last few weeks. What we need to do now is take this simple theory and apply it to the design of our experiment, okay? So what's the essential thing we need to calculate now to understand our experiment? What does this tell us we need to figure out? Yeah, we need to know what kappa is, right? That's the most important thing, okay? So let's estimate the length scale. Okay, so as we said before, I'll write the equation again. Kappa squared equals k cat phi c over km df. Okay, so it's a relatively simple formula, but it sort of drives me nuts as an experimentalist because I want to look at something macroscopic, right? But this penetration length seems to depend on the details of how my catalyst works, okay? As an experimentalist and as a physicist, I abhor molecular detail. So I want to find a way of getting a sense if how can I avoid that? Okay, so do I really need to know the detailed the microscopic info about my catalyst? And the answer is no, actually. I don't need to know about it at all, right? Because I can work backwards. And what do I mean by that? I started the lecture today reminding you that what I'm targeting is a particular metabolic rate, okay? And I know that all organisms have a metabolic rate between a kilowatt per meter cubed and a megawatt per meter cubed. So what I'll do is I'll try to work back from that metabolic rate, and then I try to express that length scale in terms of the metabolic rate instead of the microscopic details, okay? So, yeah. So this is the thing that we're going to specify, right? That we have some metabolic rate. And we know that that metabolic rate is equal to the reaction rate, okay? Or at least is well approximated that reaction rate times the change in enthalpy of the reaction that is the source of fuel, okay? So this is the enthalpy change in going from associated with converting the fuel into the waste. Okay, so this requires me to know some chemistry, right? But at least it's easier to deal with chemical thermodynamics than it is to deal with chemical kinetics, right? So yeah, so okay, so that's one little bit. That doesn't get us far enough. But the other thing I can do, as I know that as I just showed you, in the limit where my sphere, my domain is small, or my diffusion coefficient is fast, I have a homogeneous reaction rate inside of my domain. And that homogeneous reaction rate is just kcat, the concentration of the catalyst, divided by Km, times the concentration of the fuel at infinity, okay? Because in this limit, right, it's unchanged as it goes in. So I can combine these two equations and stick it into there and remove the microscopic information of the catalyst. And I get that the square of the penetration length, or the kappa squared, is just equal to q dot divided by the change in enthalpy associated with the source of heat times the diffusion coefficient of the fuel times the concentration of the fuel at infinity, okay? I like this as expression because everything is in terms of macroscopic parameters, and those are ones that make it easier for me to design things. So now I have a useful expression that doesn't bog me down in wedding myself to a particular chemical mechanism yet. But to get numbers, I do have to commit to a chemistry, okay? But before I do, what questions do you have? None, okay. Okay, so let's forge ahead. So what we're gonna do is, we've been interested in sort of physiologically relevant metabolic rates. So let's pick a physiologically relevant chemistry, okay? So we need to pick a reaction. Okay. What reaction would you pick? If you wanted to describe, if I have a fuel and I have a waste, it's driving metabolism in biology. Yeah, that's what I thought I was going to do at first too. But then I thought about how ATP actually works inside of a cell, right? So I don't have my little domain anymore, but here's my little domain, right? ATP is actually made inside of the cell, right? And ATP doesn't diffuse in from infinity to feed the cell, right? It's a local source of currency, but it's not the thing that's ultimately sort of diffusion limited from infinity, right? Does that make sense? Sugar, yeah, exactly. The next best thing, sugar. So I think sugar is a better choice, right? So we're gonna use glucose, right? So sort of there's this famous process called glycolysis, right? And it's one of the most common. I'll just say super common. In my notes I have one of the most common sources of energy in biology, but super common is enough to write on the board. And I apologize for writing a chemical formula on the board at a physics school, but I'm going to do it. So glucose is C6H12O6, okay? So here's glucose. The way it works is that it basically gets burned in a controlled manner in a process called respiration to make carbon dioxide and water, okay? Yeah, so we're going to imagine that our fuel is glucose. And then once we've done that, we just need to look up some numbers from various sources to be able to figure out what our penetration depth is. All right. So the first thing we need to know is the enthalpy of this reaction, and it's about 3 megajoules per mole. Okay, it's a number. It's big. You burn sugar, it makes a fire. The diffusion coefficient of glucose and water is about, and this is not cytoplasm or anything. This is just water. Is about 7 times 10 to the minus 10 square meters per second. And then the last thing we need to know, and these are things you can just look up in chemistry books, the last thing that sort of had me stumped for a little while was what to use for the concentration of glucose at infinity. And ultimately what I decided was to use the concentration of glucose in the blood, because this is well documented, because people measure it every time you go to the doctor and they take blood. They measure the concentration of the glucose in your blood, and if you're healthy, it's somewhere around 5 millimolar, or and that's the same as 5 moles per cubic meter. And so this, we're going to choose that value because it's the concentration in blood. Okay. So yeah, so when we these are from the doctors. This number's from the doctor's office. These are from the chemistry tables. Then you find the following values for the penetration depth. Okay. So we're going to do it at 2 metabolic rates. Of course, we're going to do it at 1 kilowatt per meter cubed, and we'll do it at 1 megawatt per meter cubed. Okay, so we expect that it's going to be a lot smaller, of course, for the higher metabolic rate. Anybody want to guess how big it's going to be? Any guess at all? Very large. What is large for you? Okay, I won't say anymore. What do you think he means by very large? Oh, that's a good guess. Because it is 100 microns uh, for this one, and it's 30 times bigger for this one, so that's about 3 millimeters. Okay? So that is good news. It's not great news, but it's good news. The reason why it's good news is that both of these length scales are much bigger than any structural scale of any active material I would ever want to make, right? So it really suggests that there is sort of a few orders of magnitude of length scale where the system was homogeneous. I can think of it as a material, and then I can start to look at the relationship between the material properties and the activity, which is something we're working on, okay? It's not great news because these are pretty small samples, okay? But they're still workably big with modern technology, right? So we can work with these. So I'm happy because this is much bigger than the structural scale, and I'm sort of like slightly happy about because it's sort of reasonable. I would say it's a reasonable scale for experiments. Right? It's something you can work with. But I can't just stick the stuff in a regular rheometer or something like that. I'd have to do things slightly more sophisticated. Okay, so that's all good. Okay, so that's the fuel side of things, and now we're going to move to the heat and the waste. If people are ready. Okay, so let's do the heat and the waste. All right, so we're not going to do heat and waste generally. We're just going to do the heat and waste in the limit of kappa r much less than 1. Because I don't even want to bother doing the experiment if I don't have a homogeneous reaction rate, right? So we'll just consider that limit. In that case, we already showed, yeah, but I've erased it, so I'll write it again, that del squared phi w is equal to this source term. But what we now know is that in this limit, this is actually a homogeneous source. So it makes solving the math very easy. So easy, even an experimentalist can do it. Okay, and then and I'm not going to go through the derivation, but I recommend that you do it yourself if you haven't done something like this before. But sort of if you just follow the classic line of reasoning, if you follow a similar line of reasoning, you get almost the identical equation for the temperature. So the Laplacian of the temperature is equal to some source term, which is now S delta H divided by rho C alpha. Rho C alpha. Okay? And I'll tell you what rho C alpha are now. So rho is just the density. C, so that's kilograms per meters cubed, right? And C is the specific heat. And that is joules per mole per kilogram. And alpha is the thermal diffusivity. And that is a normal diffusion coefficient of meters squared per second. So often you will see rho C alpha together, and when they're together, they are often called as a single material property the thermal conductivity. Because in order to derive this, you start off by saying that the flux of heat is equal to minus rho C alpha grad of T, okay? That's the thermal conductivity. Okay, so a really important thing to point out about these equations, in contrast to the governing equation which I erased for the fuel, is that these two equations, right, don't have a length scale built into them, right? So they have a fundamentally different nature. And it's clear when you look, that's clear when you look at the solution. I'm also going to mention that, sort of keep things simple, you can do it more generally. The solution I'm about to write down just assumes that the thermal diffusivity and the diffusion of the waste, the diffusion coefficient of the waste, are the same. They don't change. Not that they're equal to each other, but they're the same inside and out of the reactive domain. And again, I mentioned, I didn't say this explicitly today, I said it explicitly last time, that there's no diffusive barrier between the reactive domain and the outside world. Okay? So when you do that, you get a simple form, and because these two equations are the same equations with different variables, they have this, if you normalize things properly, they have the same functional form. Okay? So the concentration of the waste at any position compared to the concentration of the waste at infinity, normalized by the excess amount of waste at the center, and I'll write that down in a moment, has the same functional form as the temperature profile minus the temperature profile at infinity divided by the excess temperature at r equals 0. And again, I'll write that down in a second. And that functional form is like this. And again, it has two domains because the inside and the outside are different. The inside, of course, has the reaction. The outside doesn't. Inside, it's just 1 minus 1/3 little r over big R squared. And that's for inside. And then outside, it's even simpler. It's just 2/3 little r, big R over little r, sorry, big R over little r. And that's for outside of the reactive domain. And of course, you know, if I solve this properly, the fluxes at the boundaries will match, and also the values of the inner domain and the outer domain will match. And you can see when r is equal to r, this one is equal to 2/3, and this one is equal to 2/3. So at least the main scalar values match. Take my word for it, or at least check yourself that the fluxes also match. Okay, so that's a very simple form, and you can see that, you know, there's no inherent length scale in the equations. The only length scale that appears in the problem is the boundary condition. It comes from the boundary conditions, right? Here, the length scale other than little r is big R. Okay, so the system, the diffusion of the heat and the waste don't have any inherent length scale. So now I'll just sort of define what I mean by these two and put them in a useful form. So So this is the sort I call you'll become obvious in a second, the excess at the center. So it's phi w minus at r equals 0 compared to phi w at infinity. And that's proportional to the reaction rate times r squared divided by twice the diffusion coefficient of the waste. Similarly, the excess temperature at the center is, you know, T at r equals 0 minus T at infinity. And similarly, that is proportional to S times delta H over rho C, and then r squared over 2 alpha. Okay, so both of them have a form where you have r squared over a diffusivity and a prefactor that tells you either how much stuff you're producing or how much heat you're producing, or at least how much of a temperature increase an increment of the reaction drives. So yeah, so the two key observations from this solution, right, is that there's an excess of temperature and waste inside the droplet, okay? That TR minus T infinity is positive, right? So it's hotter inside of the droplet. And similarly, there's an excess of waste inside of the droplet, just as you would expect. The other important thing is that you know, the concentration of waste and temperature inside the droplet are, you know, scale like R squared. Okay, so it's quite sensitive to the size of the droplet. It's not as sensitive as the fuel was, right? The fuel was exponentially sensitive to the size of the droplet, right? There was a relatively sharp boundary between what was acceptable for fuel and what was not acceptable for fuel. For waste and for temperature, the boundary is more diffuse because of that R squared scaling instead of the exponential with R scaling. And so what we need to do to sort of get some numbers out is we're going to have to I'm going to introduce sort of a heterogeneity parameter, an amount of temperature heterogeneity that I'm happy with, and then we'll figure out how big the droplet can be to be consistent with that. Yeah. Oh yeah, so the amount of heat, so let's think about the heat because it's a little bit easier, more used to it. So you're producing heat uniformly throughout, so the amount of heat produced goes like R cubed, but the rate at which you get rid of it goes like 1 over R. And the simplest way to think about that is that the diffusive fluxes go like the gradient of the temperature, and the gradient of the temperature scales like the temperature over R, right? So, and then you, yeah, and that's how you get R squared. That's basically how it works. Thank you. Okay, moving along. Okay, yeah, so like I was saying, yeah, we have to, what we're going to do is we're going to sort of define a heterogeneity parameter, okay? So we need a simple measure of heterogeneity. This thing has no deep physical significance. It's just a number that I can use to see how homogeneous the system is. So I'll just call it epsilon W, which is my measure of how heterogeneous the concentration of waste is inside of my sample. And I'm just going to basically compare the waste concentration in the middle of the drop to the waste concentration at the edge of the drop, right? Because I want to see how much it changes over the droplet. And because I want it to be dimensionless, I will just normalize it by the waste contribution, waste at infinity. And I'll do exactly the same thing to quantify how heterogeneous my temperature is. I'll just take the temperature at the middle compared to the temperature at the edge and normalize that by the temperature at infinity. You know, and ideally, right, these things, both of them, are going to be much less than 1 if I want to have a homogeneous system whose properties I can characterize in the lab. Yeah. Yeah, I could have. I don't think it's going to change the answer too much. Oh yeah. Okay. Actually, that's sort of funny. Yeah, that's a really good point. And that's actually how I set up the calculation at first. And then two things caused me to write it this way. The first thing that caused me to write it this way was symmetry. I can always set this equal to 0, right, on all the calculations. But the other thing, which is actually a better reason than symmetry in this case, because this is not a theory of everything. This is like a rough calculation to get my experiments to work. So this should be experimentally motivated. It turns out that there's a pretty high concentration of carbon dioxide in your blood actually, and also if you're growing bacteria like in the medium, like so they're used to living with some background concentration of CO2, and so that's why I use something that was non-zero at the end of the day. Okay, so yeah, so if we say okay that if we require these don't exceed some value, then that puts constraints on the size of the system, right? Because, well, I'll show you. So we require that the size is less than, for the waste, this is going to be our threshold size for the waste, and it goes like the square root of a bunch of stuff with the metabolic rate on the bottom, 3 times my figure of merit, diffusion coefficient, concentration at infinity, and delta H. And similarly for the temperature, has almost the same form. We get 3 epsilon T, the conductivity, the temperature at infinity, divided by Q dot. Okay, so that's my criterion about whether or not my system is small enough. Okay, so to move forward, so this is nice and simple again. It's all in terms of, just like before, it's in terms of macroscopic quantities. I don't have to worry about molecular details. But in order to make a calculation, I need to wed myself to a chemistry. And so, of course, we'll stick with glycolysis to be consistent. And there, the waste in that case is carbon dioxide, okay? And in that case, the delta H is not the same. It's nearly the same. It's just 6 times smaller because there are 6 molecules of CO2 made in the reaction. Okay, so there's not 3, but 0.5 megajoules per mole of CO2. And as I mentioned, in the body, again, we're just going to keep things sort of using the levels in blood. There's actually quite a high concentration of carbon dioxide in the blood. It's 30 millimolar. And this is in blood. Interestingly, that's like 6 times bigger than glucose, right? Which I guess sort of makes sense because your body's converting glucose into carbon dioxide, and it makes 6 moles of carbon dioxide for every mole of sugar it uses, but it's sort of suspicious that it's exactly 6. Or maybe not, I don't know. I'm slightly surprised about that. And then the diffusion coefficient of carbon dioxide in water, you can look it up, and it's twice times 10 to the minus 9 meters squared per second. And if I sort of somewhat arbitrarily set a cutoff of homogeneity of 10 to the minus 3, so 10 to the minus 3 for temperature would correspond to like 0.3 Kelvin, which is, that's a pretty, I don't know if that's a small or a big temperature difference. It sounds small at least. And epsilon W for the waste, not knowing anything about how toxic carbon dioxide is, I just decided to use the same one that I used for the temperature, okay? And it's nice and small. And there's some interesting results here. I also have to give you the numbers for, this is all about carbon dioxide. We'll take the temperature at infinity just to be 300 Kelvin. I could have made a body temperature slightly higher. And the conductivity of water is 0.6 watts per Kelvin per meter. And that's water. And so I'll make a little table here. So we have 2 different values of Q dot in watts per meter cubed. We have 10 to the 3, and we have 10 to the 6th. And for each of them, we're going to calculate RT. So this is the size limit due to the temperature, and the size limit due to the diffusion of the waste, okay? So what do you guys think? Do you think it's going to be that the temperature and the waste will put more or less stringent criteria on the size of my experiment than the availability of fuel? Any opinions? Well, it turns out that heat diffuses a lot more quickly than matter does, right? So if in order for a water molecule or a CO2 to diffuse from one side of a sample to another, you know, it collides with all the other ones and it gets interrupted. But the vibrations, right, have basically, they can travel more easily, basically. And so the cutoff due to just the thermal due to heating is at the lower metabolic rate corresponding to humans is only about 2 centimeters. That's really big. That's easy to work with. And it's not relevant because it's bigger than the one I had for the fuel. And for the higher metabolic rate, it's about 1 millimeter. Okay? So both of these are bigger than the constraints that I had from the fuel, so they're not relevant. But the waste is actually a bit of an issue. For the waste, and again, I don't know how arbitrary this cutoff is for the waste. I get a constraint that the system has to be smaller than about 300 microns at the human metabolic rates, and it has to be smaller than about 10 microns at bacterial metabolic rates. So it's actually quite the getting rid of the diffusing away the carbon dioxide actually seems to actually be the rate limiting thing at the metabolic rate of a bacterium. Another interesting and important thing, you know, having done this calculation and the one before, is to compare these numbers against the sizes of cells and the sizes of bacteria, right? And so if we look at bacteria, the size of a bacteria is a micron or a few microns, right? And it really says that a bacterium, even though it has an isolated bacterium, even though it has a huge metabolic rate, is essentially at the same temperature as in its environment, and that it's small enough to be able to get rid of the carbon dioxide inside without having to do anything special. Okay. Yeah. 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 Mm-hmm. Yeah. I was actually googling that in preparation for this, and well, the Prandtl number is the one for that they use in sort of like, you know, the for the boundary layer between. They talk about boundary layers, right? So there's, if you have over short time scales, short length scales, diffusion always dominates, and then large length scales, it's you know, it's convection. And if you, so in these experiments, and this is a point I'm about to make, that I'm assuming everything's diffusion limited, right? If I convect on the outside, I can improve these numbers a little bit, but not a lot. What you really need to do to get these numbers a lot smaller is to have internal convection, which is a lot harder from an experimental standpoint. But I guess to answer your original question, I don't know. But certainly the people that do heat transfer talk about must have one, because they talk about the boundary layer thickness. Okay, so yeah, that's basically the bulk of that is all of the calculation that I wanted to do today. Okay, and so now let's just talk a little bit about what it means and where to go next. Please. Yes. Yeah, exactly. So yeah, so at the end of the day, I think what really in that particular case, what's going to matter is the narrowest dimension of your sample. And I think all these calculations in different geometries, you wouldn't really have to redo this calculation for a geometry like that. I would just use the same threshold, but apply it to the narrowest dimension of your sample. And yeah, external convection will help, but will only change things by like a factor of 2. It's not really, it's not going to make it. It's not going to solve the internal problem. You can only solve the internal problem with internal convection. Yeah. Yeah. Well, that's a great question. So let's think about that. So, I mean, there's a number of things to discuss there. I mean, I mean, I'm sure that some of the waste of a cell just diffuses freely. I bet carbon dioxide is not assisted at all in getting out of the cell, right? So larger things, and there's two things that inhibit uh, the rejection of waste inside of a cell. Um, One of them is sort of just the fact that some of this waste might have a higher molecular weight and a lower diffusivity inherently. Viscosity of the cell is higher than the diffusivities I'm using here. But perhaps more importantly, I would say, is the fact that you have the plasma membrane, right? And the plasma membrane is a diffusion barrier. And unless only the very smallest molecules can, and they have to be neutral molecules, can diffuse across the plasma membrane. Anything that can't diffuse across the plasma membrane needs to be pushed across it with a vesicle, and that is consumes ATP. So, yeah, and that's the case where you need to where you need to use energy. Yeah, and another, it's yeah. Okay, so yeah, I'm going to write down my conclusions. Okay, maybe we'll finish a little bit early, which is not a bad thing on the longest lecture day of the school. Okay, so the conclusions of this analysis, right, are that yeah. So yeah, I'll write the whole sentence. Sort of the maximum size of a homogeneous system uh, is small but macroscopic. Okay. Yay! That means we can over the full range of the metabolic densities that you see inside of cells. And one thing that I think it's fun to point out here is that these length scales that I pointed out for the limits of mass transport and of the fuel, the waste, and the not so much of the temperature, but of the fuel and the waste, correspond to roughly the density of the circulatory system. So like your circulatory, once you go down to a few hundred microns, you've got a blood vessel nearby to advectively take away your waste and provide you with fuel. So once you can rely on diffusion for the last 100 microns or so, or a few hundred microns, but you can't really rely on diffusion much beyond that. So that's sort of, these numbers are not just relevant for me figuring out how to do my experiments, but they're also help make sense of the way our bodies are built. Okay, and so typically, mass diffusion, mass fluxes are limiting, because even for the smallest species, the thermal diffusion coefficient is much bigger than the mass diffusion coefficient, and so generally you have a problem with mass transport before you have a problem with heat. The other point, which I've said a few more times, so I won't write it down again, is that this maximum size is also much bigger than the structural scale, so there's a few decades available for continuum mechanics. And of course, we've already said it, that you can do a lot better with a circulatory system. Right, and that's why we have a circulatory system. Yeah. So anyway, I won't write that down. Yeah. I was recently looking into this actually, about 9 months ago, and there's lots of good sources. You can look at Darcy Thompson, and it a little bit depends on what you mean by a circulatory system. So you have to get to a pretty large organisms, what do we call vertebrates, that before you start seeing a heart, which has to actually actively, like, there's a pump that pushes everything through. But even millimeter scale and submillimeter scale insects have a network of tubes inside of them, but there's no pump. And the idea is just as the insects just through bending and moving their bodies actually drive a convective flux. So yeah, I think it's about it's about a millimeter, surprisingly. Yeah, not so surprisingly. Yeah, tell me more. Yeah. Okay. Yeah, what's the physiological function of them? Are they okay? Mhm. Okay. Yeah. Yeah, and again, I mean, so, here I'm looking at the diffusion of small molecules. Big molecules are going to have significantly lower cutoffs. Cytoplasm has a much higher viscosity than water, and so that might be the reason why these things are popping up. I think Raman had the next question. Okay. Yeah. 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 Yes. Yes. Yep. Yeah. And of course, within a cell, there's the microtubules, which are a form of convective transport. But the thing that's about microtubules is that it's not so much they're needed to keep things mixed, but they're more involved in directed transport. So we have some small copy number thing, and we want to move it in the cell specifically from here to there, and that's where microtubules work really well. They're not just trying to mix things up. Yeah. How wedded am I to water? Yeah. Yeah. In sort of in reality, the systems that we are looking at are, they're all aqueous-based systems, but like cytoplasm, are dense polymer solutions. So we typically use dense polymer solutions, actually. It would change it by a factor of the viscosity, and dense polymer solutions can have, orders of magnitude varying viscosity depending on the details of the solution. So it could be a disaster or it could be fine. Yeah. So anything else? Okay, that's it for today. Thank you.